Thank you. Yeah, so I love introducing the brainstem because it's super easy. I don't really have to say anything. We all know what it is. We know it's important. It keeps us alive. Unfortunately, it's also one of our favorite, or at any rate, one of my favorite brain regions to ignore until now, or to represent just as a single region. And a lot of what we know about brainstem functional neuroanatomy comes from animal studies, human brain specimens, tends to be specific to specific nuclei, specific pathways, and very histology dependent. So what this means is what we're really missing is this whole brainstem in vivo understanding of what is happening functionally in the structure in a human that's awake and also preferably, well, alive and preferably awake, I guess I should say. So for this project, I'm not just focusing on the brainstem. It's very much about the interplay between the brainstem and the cortex. We spent a lot of time looking at the cortex in terms of its functional architecture and now what's going on in the brainstem as well. There are a few reasons that brainstem functional imaging has lagged behind cortical functional imaging, one of which is that we need really high spatial resolution and high sensitivity to image these really deep nuclei that are also extremely small. We need an atlas or a template of brainstem nuclei. If you're just interested in one brainstem nucleus, then you could probably go and delineate that nucleus yourself and study that. But if you want to do the whole brainstem, that is a lot of work to figure out where all those nuclei are. So having some sort of atlas would be really helpful. And very important, physiological noise reduction pipelines. As you know, the brainstem is hanging out really close to CSF, really close to vasculature. Did you know every time your heart beats, your brainstem wiggles? So you can just imagine how difficult it is to get good clean signal there. That's where Marta Bianchardi comes in. She's a professor at Harvard, and over the past almost 10 years now, she's been really carefully delineating brainstem nuclei in MNI 152 template space. And very recently, her group put out this functional connectivity between brainstem and cortex, and this is functional fMRI data that's specifically acquired for the brainstem. So this is the data I'm working with. Here's the brainstem there. I'm mostly going to plot it as node centroids, just a little bit easier to see. And what you need to know is there are 58 brainstem nuclei in total, 400 cortical regions that I'm looking at, and this brainstem-specific data set is acquired in 20 people. One of the first things we do is just make sure that the atlas of the brainstem nuclei maps on as what we would expect from receptor densities in these different nuclei, and it does, that checks out, so that's good. So here's the functional connectivity data. And you can see cortex to cortex functional connectivity is quite a bit bigger than anywhere in the brainstem, probably due to SNR. But if we do zoom in on cortex to brainstem functional connectivity, we see these vertical and horizontal stripes. And these stripes indicate that there are dominant patterns of cortex to brainstem connectivity. So what are those patterns? So to figure that out, what we do is we take for every brainstem nucleus, we sum across the cortical regions the functional connectivity with the cortex, and we just plot that on these node centroids here. So every node here is a brainstem nucleus, and the color and size is telling you how much this brainstem nucleus is functionally connected with the cortex. What I like about this network is if you imagine you want to make a network that is really efficient in terms of its information and communication capabilities, you would probably put the hubs at the top, in the middle, and at the bottom. And that's what we see here. We see the hubs of brainstem to cortex connectivity at the top, in the middle, and at the bottom. On the flip side, you can do the same thing for the cortex here. The question is, which cortical regions are more functionally connected with the brainstem? And here we see this pretty strong anterior to posterior gradient. And this gradient actually aligns really well with the cellular and structural architecture of these regions. So we find that paralimbic and insular regions have the most functional connectivity with the brainstem. Okay, so one of my main questions that I wanted to address when I first got this data was this: do groups of brainstem nuclei make distinct connectivity patterns with the cortex? And so another way of saying that is, can we cluster the brainstem in terms of the way that it's connected to the cortex? So here's a brainstem by brainstem similarity matrix. This is telling me how similar are two brainstem nuclei in terms of the way that they're connected to the cortex. And if you order the brainstem nuclei just so, you can see that we form these clusters of nuclei that are really similarly connected to the cortex, not so similar to the other communities, but very similar within. Here I'm just plotting those community labels, so I get 5 different communities, different groups of nuclei, they're spatially segregated, and then for each of these I look at how are those nuclei connected to the cortex. So just to give you an example, how are the yellow brainstem nuclei functionally connected to the cortex? Well, that would be this cortical pattern below the yellow bar. And what I like about these cortical patterns is as soon as I saw them, they were very recognizable. I could sort of see the networks that are coming up here. For example, that yellow one is very somatomotor, the pink one is very cingulate. And you don't have to take my word for that. We did a neurosynth decoding just to see which functions are related to these different cortical networks. And indeed, it's very functionally relevant and also very kind of recognizable networks, things related to perception, things related to action, things related to emotion regulation, anxiety, but also more higher order cognitive functions and networks as well, which was kind of surprising because this is the brainstem. So then the question becomes, well, okay, why are these brainstem nuclei connected to those cortical networks? And a sort of maybe obvious direction to go in there would be to look at neuromodulatory systems, because we know that there are all these receptors being expressed in the cortex, but also that the brainstem is housing a lot of these nuclei that are synthesizing the neurotransmitter. So we did a multivariate mapping between receptor densities and these cortical networks related to brainstem connectivity. And indeed, we find this three-way relationship. I'm just going to give you some example. For the dopamine transporter ends up being the most dominant transporter for the green community. The green community is where the substantia nigra is. Substantia nigra, that's where lots of dopaminergic neurons are. Another example, the norepinephrine transporter, most dominant for the blue community. This is where the locus coeruleus is. That's the nucleus for noradrenergic neurons. And so this was kind of a cool link between which brainstem nuclei are connected to which cortical networks and why. Okay, so all of that has been all about how are brainstem regions similarly connected to the cortex. Now the flip side of this is how are cortical regions similarly connected to the brainstem. And if you recognize this parcellation in Schaefer 400, and then take the first gradient of that, you'll see this very, by now famous gradient, this anterior to post, this unimodal transmodal gradient. And so yes, indeed, it is very much correlated with the functional gradient you get from cortico-cortical connectivity, but very importantly, this is not cortico-cortical connectivity, this is cortex to brainstem connectivity. And one difference between these two gradients is that the cortex to brainstem gradient is quite a bit more bimodal than the cortex to cortex gradient, despite the two of them coming from the same method, diffusion map embedding. And so I sort of hypothesize or wonder if this means that brainstem input seeds the two extremes of this gradient, the transmodal extreme, the unimodal extreme, whereas cortico-cortical connectivity kind of fills in the shift in between. And then the question becomes, well, where in the brainstem are unimodal brain regions connected, and where in the brainstem are these transmodal brain regions connected? And we see that unimodal regions are mostly connected with caudal brainstem plus the inferior colliculus, and that transmodal cortical regions are mostly connected to rostral brainstem regions minus the inferior colliculus. And this checks out with literature as well in terms of what these nuclei are doing. So in summary, we have this 70 fMRI brainstem data set, plus this whole brainstem atlas, and this lets us expand our perspective of the connectome from just the cortex to the cortex and the brainstem. We identify hubs that represent high connectivity between the brainstem and the cortex. We find these clusters of brainstem nuclei that are related to very well recognizable and functionally relevant cortical networks. And we investigate what is the link between those brainstem nuclei and those cortical networks, and we find it's probably to do with the neurotransmitter systems that are both in the brainstem nuclei and also being expressed in the cortex. And finally, we find that this unimodal transmodal cortical hierarchy, which typically is understood as a gradient of cortex to cortex connectivity, also represents cortex to brainstem connectivity. So if I were to sum this up in a sentence, I would say that a lot of what we see in cortical function can be traced back to the brainstem. A lot of the things that we think of as purely cortical, I think, are also appearing in the brainstem. So that's all for me. This work is already pre-printed, so it's up there. Big thank you to the co-authors, the collaborators here, especially Marta Bianchardi and her group for acquiring this data and sharing it with me. Code and data is on GitHub, and I will have a poster tomorrow and Thursday. Thank you.
For that wonderful talk. So we have a couple questions. When comparing brainstem cortex connectivity with neurosynth functional maps, how do you account for spatial autocorrelation? So is there some sort of general signal that you're picking up, or how do you know it's specific?
Yeah, for sure. A spin test. Yeah, baby. So I'm just taking for each brainstem nucleus, I'm looking at the way it's connected to the cortex. That's going to give me a cortical brain map. And then for each neurosynth map, that's a cortical brain map too. Those are just being correlated with one another and spin tested, as always. Yeah.
Perfect. And that also answers the second question. But I do have another question. Given that there's a lot of different concerns kind of about SNR and signal to noise ratio in different parts, how do you kind of account for that in the brainstem, especially given that they are such small nuclei?
Yeah, definitely. So pretty much we try to go at this from many different directions. One thing we have is the temporal signal to noise map. So every time I did something, I just correlated with that just to make sure that this is not just temporal signal to noise. But I'm sure that SNR is playing an impact. Like you saw, functional connectivity is lower in the brainstem. Maybe that's because most of the connectivity goes out of the brainstem, which I think biologically makes sense. But it could also just be because SNR is quite a bit lower. Yeah.
Great. Thank you so much.
Thank you.